Alright, so far what we have done is the generalized structure of cartilage, the basic structure of cartilage. We also started off with basically the anatomy of cartilage and where it's present and how it looks like, yeah? And then we defined some of the general structure of cartilage on a histology slide, its general structure molecularly, right? We defined it through a diagram, then we looked at some images as well, and we said that basically cartilage is a tough and durable connective tissue which has a frictional surface, and what it contains is cells enclosed in lacunae, and those cells are called what? Chondrocytes, right? So chondrocytes are the only cell type present generally in cartilage, and cartilage is surrounded by a dense connective tissue layer called the perichondrium, right? And that perichondrium contains progenitor-like cells that divide to form chondroblasts, right? Chondroblasts, and then those chondroblasts divide to form chondrocytes, right? And those are the cells that are present inside the lacunae of cartilage. Okay, so that was the general structure of cartilage, but now we are going to go into the specifics of the different types of cartilages. So when we started with this chapter, we defined that there's actually 3 different kinds of cartilages, right? There's hyaline cartilage, there's fibrocartilage, and there's elastic cartilage. So now we are going to learn the differences between these cartilages, what makes them suited towards their function, where they're present, how they look on a histology slide, and most importantly, we will try to learn to differentiate all of these different cartilages from each other on a microscopic slide. So hopefully by the end of this lecture, you will be able to distinguish which type of cartilage you're looking at on a histology slide, okay? Alright, so let's get started with the cartilage number 1. That would be the hyaline cartilage, which is the most abundant cartilages that is present inside the human body. So let's start with its anatomy and where it's present inside the human body. So in this diagram as well, I'm going to write the anatomical locations down over here, but basically it's present in nose, of course, and then it's present in the C-shaped rings of trachea, right? And it's present inside the respiratory tract, and it helps keep the respiratory tract open. Another place where it's present is the costal bones. At the end of costal bones, it's got the costal cartilage, and you must have studied this in anatomy. So we have costal cartilage at the end of costal bones, right? And it is also present in synovial or articulating joints or sliding joints, synovial joints, and it helps the bones to articulate with one another, right? Another place that it's present in is, we will study more about it in the next video, but basically it's present inside the epiphyseal plates of long bones. So for example, let's look at this long bone right here, which is the humerus. I'm going to dictate this here. Yeah, this is the humerus, right? And you might have studied it in the anatomy, but there's an epiphyseal plate right here. This is the epiphyseal region. In growing bones, this epiphyseal region is made up of cartilage, okay? So it is made up of cartilage, and that cartilage is hyaline cartilage. So hyaline cartilage is present also in the epiphyseal plates, and what it does is that it allows the bones to grow. So it's got these cells inside the cartilage, and these cells, as they grow, they help the elongation of bones, and then that cartilage is then ossified or basically calcified into a tough, strong bone, and we will study the details of it in the next chapter. But I'm going to write this down, even though we will do it in the next chapter. It's called elongation of bones. Elongation of bones at epiphyseal of long bones at epiphyseal plates. Okay. Okay, so let's go on to the specific features of cartilage, what it looks like molecularly, and I'm going to use some of the photos over here. I'm going to start with this photo right here, as it looks pretty good when you see it. So basically this looks very similar to the generalized structure of cartilage, right? It looks very similar to a general structure of cartilage. So what it contains is, okay, this area which is very, very pink, and it's got these elliptical cells, right? It's got these elliptical cells. These are not completely round cells. These are elliptical, and it is a very pinkish tissue, which is obviously a dense connective tissue, and we've studied it. It is the perichondrium, right? And this perichondrium, and then it contains is elliptical cells that are basically the chondroblasts, and then these chondroblasts mature and form these mature circular cells, now not elliptical anymore, circular, and these circular cells are called chondroblasts. Now what I want you guys to notice here is that the extracellular matrix that's outside of the lacunae, it is very, very clear, and it is pretty much the same everywhere in the cartilage, so we could say that it is homogeneous. The ECM is ECM. The extracellular matrix is homogeneous, and this is a very key point to remember, that it's homogeneous, it's the same everywhere, it does not vary with location. It's pretty much the same pinkish texture that it has, and it does not have any basically changes going on in there. It's very, very homogeneous ECM. And the way it's colored so clearly is because it contains, basically, we studied that earlier in the general structure, is that it contains proteoglycans, and one of them is proteoglycans, right? And one of them, the most common in cartilage, is aggrecan. Aggrecan is a proteoglycan. What are proteoglycans? We studied that in connective tissue, so I'm not going to go over it over here, but it contains one of the most common proteoglycans in the body. It's called the aggrecan. And what this aggrecan is able to do is that it is able to bind to hyaluronic acid. Okay, this is a very water pulling GAG. What it does is that it pulls in the water. It allows the water to come towards it. It's very water loving, and it basically pulls the water towards it. So because it's present inside the cartilage, right? It is bound to the aggrecan. It is present inside the ECM of the cartilage. In this case, we're studying hyaline cartilage, right? So it's present in the hyaline cartilage. Then what it does is that it allows lots and lots of water to come in to the ECM of the hyaline cartilage, right? And this results in water forming the major component of the extracellular matrix. It's actually around, if it's a fresh hyaline cartilage, it's around 60 to 80% of the mass of the hyaline cartilage is formed by water, because it's been pulled towards the hyaluronic acid, which is very water loving and loves to pull the water in, and is bound to aggrecan. So basically aggrecan is the proteoglycan that allows the binding of hyaluronic acid, which pulls in the water. And as aggrecan is basophilic, so let me just write that down. Aggrecan is basophilic, so basically the ECM is also basophilic, right? Two other GAGs are also present. So one of the GAGs is hyaluronic acid, which is present, and it is bound to this proteoglycan that is aggrecan. Another GAG that is present is chondroitin sulfate. Tin sulfate. And another one is keratin sulfate. These two GAGs are also very abundant in hyaline cartilage. So in case of people who have a wear or tear of cartilage, especially wearing off of cartilage, usually seen in arthritic patients, especially in old age, when with age the cartilage disintegrates. So for example, you have some articular cartilage between basically bone joints, and it disintegrates due to aging, then sometimes they are given the supplements of chondroitin. So these are, I'm going to draw this in green. Supplements of chondroitin are given together with maybe some keratin, but chondroitin, and there's also something called glucosamine. The supplements of chondroitin and glucosamine can be given to treat patients who have arthritis due to basically age-related age-related wear and tear of the articular cartilage. Alright, so let's go to this one diagram that I want to show you. I want to show you what's happening inside the extracellular matrix. We studied what's inside the extracellular matrix. We said that it contains proteoglycans, different kinds of proteoglycans, and also contains different GAGs, right? And one of those GAGs is hyaluronic acid, which is right here. Hyaluronic acid is bound to aggrecan, which is one of the major proteoglycans present inside the hyaline cartilage. So some other GAGs that it contained was chondroitin sulfate and also keratin sulfate. Okay. So what the ECM contains is also type 2 collagen. Okay, so hyaline cartilage has type 2 collagen. So the cartilage, the hyaline cartilage that you see, for example, in the costal bones, inside the articular joints, inside the trachea, inside the respiratory tract, all of those cartilages will have what kind of collagen? Type 2 collagen, right? They have type 2 collagen. And what that type 2 collagen, where is that type 2 collagen present? It is present inside the extracellular matrix, surrounded by, or it is basically surrounding the which cells? Chondrocytes, right? Now this type 2 collagen is attached to proteoglycans and glycosaminoglycans, GAGs. The proteoglycan we studied, it was aggrecan, which is the major proteoglycan that is present, and it has some different GAGs like hyaluronic acid, chondroitin sulfate, and keratin sulfate. Okay, you don't need to remember this precise structure just for the sake of it, but what you need to know is that hyaline cartilage contains type 2 collagen fibrils inside the ECM, and that type 2 collagen fibril is linked to hyaluronic acid, keratin sulfate, chondroitin sulfate, basically GAGs and proteoglycans, and they end up pulling lots and lots of water towards the cartilage as well. All right, so let's move to the picture identification. I'm going to basically review what we have learned in the pictures, and hopefully you will have a very good idea by the end of these pictures of what hyaline cartilage microscopic slides look like. All right, so let's focus on this image right here. Image right here, immediately you should be able to identify that it contains, number one, it contains a dense connective tissue on the peripheries, and this is the perichondrium. Okay, now what I'm going to show you is that the perichondrium actually has two layers. It has a top layer and it has a bottom layer. And I want to mention another fact here is that perichondrium is not always present in every hyaline cartilage. No, perichondrium is there's two exceptions to the presence of perichondrium. Perichondrium is not present. Perichondrium is not present where? Any guesses? Perichondrium is not present in articular joints. Articular joints and epiphyseal plates. Okay, epiphyseal plates are the plates of growing long bones, and articular joints, we know what articular joints are, and perichondrium is not present in the articular joints or the epiphyseal plate. So what I was going to say is that perichondrium right here contains two layers. It has two layers, the top layer and the bottom layer. The top layer usually contains connective tissue elements. So this layer right here, it would contain connective tissue elements. And what are those? Any guesses? What's present inside the dense connective tissue? Well, in this case, the collagen that is present is type 1. So type 1 collagen. And what kind of cells are present inside connective tissues? Fibroblasts, right? So we have type 1 collagen and fibroblasts that are present inside the top layer. The top layer that contains fibroblast, while the bottom layer has the same collagen, but it has both type 1 and a little bit of type 2 collagen. But the cells that it contains are the fibroblast like progenitor cells, or we could say mesenchymal stem cells that basically then differentiate to form these cells, and these cells then differentiate to form elliptical chondroblasts. Right here, as you can see, these are elliptical shaped, containing lacunae. So these are the chondroblasts. And then these elliptical cells mature to form circular cells right here. So this would be a circular cell. And these are the chondrocytes. Okay, mature chondrocytes. So what these chondrocytes are able to do, and even when they're chondroblasts, what they're able to do is secrete. I'm going to focus on this little cell right here, this cell right here. What they're able to do is, let's just zoom this in. What they're able to do is, when they're mature, they're able to secrete the components of the extracellular matrix. And we studied the components. We studied that some of the components are proteoglycans, aggrecan, right? And some of the GAGs like chondroitin and keratin sulfate and hyaluronic acid. So they're able to secrete these proteoglycans and GAGs, the area in which they secrete, that is right next to the cell, just outside of the cell, becomes very, very basophilic, because it basically gets accumulated with lots and lots of proteoglycans. It becomes very, very basophilic, and it appears on a histology slide as a lacunae. But what it essentially contains is lots and lots of proteoglycans, which make it very, very basophilic, so it does not get dyed as well as the other components of the extracellular matrix. So this lacunae is very, very basophilic, because this cell right here is secreting lots and lots of basophilic components, proteoglycans. And this lacunae, this area that is basically not stained well, this is called the territorial matrix. Okay? So this area is called the territorial matrix, which is not very well stained, because it's very, very basophilic, because why is it basophilic? It's because the chondrocytes are secreting lots and lots of basophilic stuff into this territorial matrix. So the question arises as to what is the area called that is not so much basophilic, which is stained at least better than the lacunae. Well, that area, for example, right here, all of this area, right, of the ECM, this area, all of this is called intraterritorial matrix. Okay. So what you would call is this interterritorial matrix. Okay, interterritorial matrix. This area, the lacunae area, is called the territorial matrix, and the rest of it is called the interterritorial matrix. The area around the lacunae, the territorial matrix, is not stained that well, because it is very, very basophilic. Why? Because the chondrocyte that is present inside it is secreting very, very basophilic proteoglycans into it, right? But the interterritorial matrix is not so much basophilic, and therefore it is stained almost homogeneously in hyaline cartilage. Now, one thing that you might notice here is that these chondrocytes are present in aggregates of cells, right? These are present, for example, in pairs right here. If you notice this pair right here, or this pair right here, actually just scratch this one for now, maybe this pair right here. If you see them, they're present in aggregates, and these, let's go on this one right here as well. So these are present in aggregates as well, and you notice this on the slides, and that is absolutely correct, is that these are actually in fact present in aggregates sometimes, and these aggregates are called isogenous aggregates. Okay, these aggregates are called isogenous aggregates, and what's happening here is that as the progenitor cells form the chondroblasts, and then the chondroblasts form the chondrocytes, the immature chondrocyte form isogenous aggregates. So they collect against each other, and they basically exist as groups, and these groups help support the maturing cells. So after they mature, they leave this isogenous aggregate, leave this group, and become isolated. Okay, so again, in this photo as well, we see there's groups of chondrocytes, and these are called isogenous aggregates, and then these isogenous aggregates, when these chondrocytes mature, then they become isolated and exist as independent cells in their own lacunae. Okay, now immediately you should be able to see that there's a P labeled perichondrium right here, and this perichondrium contains fibroblasts-like progenitor cells that are present forming chondroblasts that are elliptical in shape. Okay, elliptical in shape, and then these chondroblasts basically go ahead, mature, differentiate into cells of the isogenous aggregates, and then they mature into cells of the hyaline cartilage chondrocytes. Now, another thing that you might see here is that the ECM is homogeneous. Okay, it does not have any variations to it. It's homogeneous ECM. And we said that the extracellular matrix, or the cartilage itself, does not contain any blood vessels. So the nutrients diffuse through diffusion. Basically, they come into the cartilage. So diffuse from the perichondriums. Perichondrium is the one that is supplied by capillaries and blood vessels, and the nutrients are released in the perichondrium, and they diffuse into the chondrocytes and help them grow. Now, the growth of chondrocytes can be stimulated, actually, and it is sometimes, in certain cases, it is regulated as well. It can be stimulated by growth hormone. You might have studied it. It's called somatotropin. You might have studied it in physiology, but it's called somatotropin. I'm going to call it growth hormone just for making it easy. So it's called growth hormone, and it is secreted by the anterior pituitary gland in response to growth hormone releasing hormone, which is released by hypothalamus. So basically, hypothalamus releases this hormone, growth hormone releasing hormone. It comes to the anterior pituitary, and anterior pituitary releases growth hormone. Now, this growth hormone does not directly affect the chondrocytes. What it does, it goes into the liver, okay, and in the liver it causes the release of somatomedins. So this growth hormone is called somatotropin. It goes to the liver and releases somatomedins. And these are also called insulin-like growth factor. And what they do is then these go into the perichondrium, and these are released by the blood vessels, and through the perichondrium they diffuse into the cartilage, and they stimulate the chondrocytes to start growing within their lacunae by mitosis, as well as they stimulate the cells inside the perichondrium to differentiate and make more chondroblasts and chondrocytes. All right, one thing I forgot to mention here is that the chondrocytes, they have a very eosinophilic cytoplasm. So they have an eosinophilic cytoplasm, which results in this very densely stained nature of them. They have an eosinophilic, so these are eosinophilic cells, and the territorial matrix that they form around them, right? This territorial matrix is basophilic, so it does not stain well. Again, this, for example, right here, the cell would be very, very eosinophilic, and this territorial matrix around it would be very, very basophilic. So it would not stain well, but this interterritorial matrix would be a mixture of both, so it does stain, and it is very, very homogeneous. Okay, so these chondrocytes, they can basically, the growth of cartilage results in the production of more and more chondrocytes, right? And more chondrocytes would result in production of more and more ECM, as well as obviously chondrocytes. So where do these chondrocytes come from? Well, we have studied one of the mechanism, right? They come from one of the ways through which they come from is the perichondrium, which contains basically mesenchymal stem cells or progenitor cells that form chondroblasts, and then those chondroblasts divide to form chondrocytes. Similarly, they can also undergo what's called mitosis, and everyone pretty much, if you are in medicine, you know what mitosis is. It is the replication of a cell, right? So mitosis. Mitosis can also occur in the cells of the cartilage, especially inside the lacunae, and especially when it is stimulated by somatomedins. So somatomedins come in, they diffuse through the perichondrium into the cartilage, and then they stimulate these cells to grow, and as a result, they undergo mitosis. And because of mitosis, they replicate into two and help the growth of the cartilage. So that's pretty much it for what is present inside the hyaline cartilage. So let's have a quick review of these photos. So we, in the first photo right here, right here, this photo, we see a perichondrium that is a dense connective tissue. We see some elliptical cells that have a small lacunae, and those are called the chondroblasts. They mature into these large lacunae containing cells called the chondrocytes. Similarly, over here, what's labeled in P is the perichondrium, and there's aggregates of cells of chondrocytes, and those aggregates are called isogenous aggregates. And we can clearly see that it has a very homogeneous extracellular matrix. Similarly, in this case as well, we have a perichondrium on the peripheries, and this perichondrium contains some elliptical cells, and these elliptical cells are producing more and more chondroblasts, and then they mature into these chondrocytes that are present inside their own lacunae. And almost immediately we can see that the basically ECM of this cartilage is very, very homogeneous. It does not have too many variations to it. And the ECM, it contains what? It contains proteoglycans like aggrecan and some GAGs. And which type of collagen is present in this ECM? Type 2 collagen is present in the hyaline cartilage. Okay, this is another slide image which contains again cells enclosed in lacunae inside the intraterritorial matrix, which is basophilic, and these cells themselves are very densely stained because these are eosinophilic cells. Eosinophilic cells surrounded by a basophilic lacunae surrounded by a matrix, which is the interterritorial matrix that is stained here, and this is a very homogeneously stained territorial matrix. And again, it is surrounded on the peripheries by the perichondrium. Now, one thing that I want to mention here before we move on is that this perichondrium there's two exceptions to it. This perichondrium is not present in cartilage of articular joints, not present in articular joints, okay, and it is also not present in growing long bones. So it is not present inside the long growing bones. The epiphyseal plates of long growing bones does not contain any perichondrium. Okay, that's about it for this chapter. Let's look at the neat notes and see what we have studied. Okay, so what we have learned is the locations of hyaline cartilage, its matrix and its components, the diagram. Remember, we looked at this diagram and what it contains, and then we looked at some histological slides as well. Then we studied about the cells, the chondrocytes. These are very eosinophilic, and they may exist as aggregates called the isogenous aggregates, and they are not very mature. And they are targeted by growth hormone to release secretions as well as to grow. And then the perichondrium, which is vascular for nutrition and exchange, and it is present in all cartilage except in articular joints and epiphyseal plates as well, which is not mentioned here. And that's pretty much it for the hyaline cartilage.